As we discussed on Monday, when you look under the microscope, you focus on resolution, you focus on magnification, and you also focus on contrast. And since microbial cells consist mostly of water, contrast between microbes in the background can be quite lousy. In order to increase the contrast, we often stain the specimens. Okay, I'm not going to focus that much on the wet mount, which you already did, so you can consider yourself experienced wet mounters. Another problem with microbes that you have seen is that when they're alive, they move. Especially if you look at something like Paramecium, some protozoans that can swim by themselves, trying to catch them in the field of view and keep them in the field of view is the task by itself. Amount of bacteria that you placed in the wet mount was quite considerable, so it was easy. You're going to see something, absolutely. But if you have just several cells and they flagellated and they swim, then it may be a little bit problematic. So in order to stop cells from moving, you have to kill them. And the process of killing cell while preserving its structure is called fixation. So you fix the specimen. You can fix the specimen using chemicals, but we're going to use temperature. It's not going to be designed like that. We're not going to use the heating plate. Instead, we're going to use the simplest heating device, the flame. Essentially, you're going to have flame, you're going to have your slide with a cover slip on it, and what you do, you pass it through the flame. 1, 2, 3. If you overheat your specimen, I don't want to scare you, if you overheat your specimen, cells are going to explode and it's going to be a huge mess. It may not be successful first time. We will do, at a certain point, we will start doing fixation, okay? It's pretty easy, especially when you do simple stains. You do this stain, you look under the microscope, okay, they're all dead. I'm going to try another one. Okay, you can do 1, 2 and see what's going on. Maybe heat it up a little less. Does that make sense? It's all trial and error. It's just the experience moment. Now, when we fix it, we stain it. There are two major types of stains, simple and differential. Simple stains will color all microbial cells the same. Whether they gram positive, gram negative, acid fast, they're all going to be, say, blue if you add crystal violet to them, or red if you add safranin. Differential stains allow you to distinguish different types of microbes or see different aspects of microbes. Simple stains can be divided into two large groups. Positive stains, the ones that stain the cell and leave background unstained, and negative ones that do not attach to the cell envelope but color the background. Positive stains are usually positively charged. The molecules of the dye are positively charged. Since they are positively charged, they can attach to the negatively charged surface of the cell. That's why they stain the cell. Negative stains are negatively charged. So negatively charged dye and negatively charged cell surface are going to repel each other. Does that make sense? Electrostatic repulsion. So stain will remain in the background. It will give you a lot of contrast, but the problem, you're going to see pretty well the outer structure of the cell if you use a negative stain, but other details are going to be pretty obscure. Differential stains, the ones that we're going to talk about are gram stain that distinguishes bacteria based on the structure of the cell wall, acid fast that allows you to identify mycobacteria, capsular stain, stain for special outer structure that is present in some bacterial cells that is called capsule, flagellar stain, obviously staining flagellum, and I think and the spore, but it's not here on the list, you will see it later. Now simple stains, often also divided into the basic and acidic. Basic stains are usually positive. Acidic stains are usually negative. Any questions on the stains? Not yet, I suppose. So, simple stains are simple. Positive, negative. No difference between the cells, no matter what the cells are. So gram-positive Staphylococcus and gram-negative Salmonella both going to look blue under those simple stains. Gram stain, on the other hand, that was invented by Hans Christian Gram, I think he was German, allows you to distinguish two major classes of bacteria. Gram-positive, that is stained blue after the gram stain, and gram-negative, that is stained red. Why it's so important? I don't know if Dr. Gram had any idea about the structure of the cell wall at the time he invented the stain. But it turns out that gram stain, gram-positive and gram-negative microbes are fundamentally different in terms of the cell wall structure. And that difference in the structure of the cell wall defines the susceptibility, sensitivity to antibiotics, sensitivity to cleaning agents, ability to cause certain symptoms such as fever, right? So that is a very fundamental property of those cells. Does that make sense? Great. So what is the process? I don't expect you to memorize this process right off the bat. Like we went through this slide and you know that. We're going to do gram staining on a separate occasion, and you're probably going to do 2 or 3 times in order to get it working, in order to get it right. So it's pretty fast method, although it looks really complicated. Basically, gram stain is based on a property of gram-positive microbes to retain the color, while gram-negative microbes do not retain the color. So how it works? First you add primary stain, which is crystal violet, and crystal violet will stain both gram-positive and gram-negative microbes the same. You're going to be violet or blue, okay, dark blue. What you do next, you add so-called gram iodine, the component that is known as mordant. Iodine makes crystal violet to stick to the cells better, stick to the cell wall better, okay? Cells are still blue. It turns out iodine makes crystal violet to stick to gram-positive cell wall, but not so much to gram-negative. So when during the third step you apply alcohol, alcohol will wash crystal violet off. It will wash it away from gram-negative cells. That makes sense? Gram-positive will keep it in. So this is your retention step. So after applying the decolorizing agent, which is alcohol, gram-positive cells will still keep crystal violet on their surface, in their cell wall, while gram-negatives will lose it. The last step, you apply safranin. It's a red dye, and these cells are already blue. They're not going to get any other stain. Gram-negatives will acquire safranin and will appear red. If you would look at this picture right here, this is gram-positive and gram-negative stain. It's a gram stain of a sample that contains the purple Staphylococcus aureus and red, well, we claim that it's red, but it's a quality of micrograph, red E. coli. Does that make sense so far? We're going to talk about the gram stain over and over again. In our age of sophisticated diagnostic technologies, fancy machines that, it looks like you bring the sample of patient's blood or sputum or saliva, whatever, stick it into some sort of a robot, and robot will tell you what microbes it has. Turns out one of the primary diagnostic tools still is gram staining. So you have a patient sample, you cultivate it on the plate, you stain it, and you already have the result whether the patient has gram-positive or gram-negative microbial infection. And that will define further the course of antibiotics that patient will take. Does that make sense? That's really, really important. It's not something that I make up to make it sound fancy. Actually, I almost cited, not verbatim, but pretty close, the words of the lady who's a head of clinical diagnostics at one of the major hospitals in the US. She says that gram-negative, gram-positive staining is still one of the major tools that clinical lab will use. Other stains, differential stains. Mycobacteria, like Mycobacteria tuberculosis, are different in their cell wall structure from gram-positive and gram-negative. In order to precisely identify them, acid-fast stain is used. The basic concept is the same. Its ability of Mycobacteria retain the special color after decolorizing agent is applied. In this case, the dye is carbolfuchsin and decolorizing agent acidified alcohol. It's just different reagents, but the idea is the same. You can see the acid-fast Mycobacteria on this slide, colored red. Okay. Capsule staining allows you to identify capsular structures, capsule layer that surrounds bacterial cells. You can see the whitish layer that surrounds those blue cells. Capsule is a protective structure. It prevents or decreases the chances for phagocytosis of microbial cells. So phagocytosis, which cells do it? Which cells of the human organisms do it? Phagocytosis. Phagocytosis. Phagocytes. Phagocytes or macrophages, immune cells, right? So if your bacteria like this bacillus is protected, or to some extent, against your own immune system, will it be more or less pathogenic to you? If it's protected against your immune system, more pathogenic. Does that make sense? Your immune system have less chances to kill it. So bacteria can survive for longer. Does that make sense? You see? So capsule makes bacteria surviving in your body longer. So it has higher chance to cause the disease. so it is more pathogenic. Now, every aspect of bacterial cell, every protein, every structure that makes it more pathogenic is called a virulence factor. For example, capsule is going to be a virulence factor. Antibiotic resistance is going to be a virulence factor. We clear? Endospore staining. Endospores are structures that have nothing in common with the spores produced by mushrooms that you can see. You know, like when you kick the mushroom, there's a cloud of little tiny particles. Endospores are special bacterial structures that protect genetic information of bacteria from destruction in dry conditions. It's kind of an escape pod for bacteria. Okay? Bacteria form endospores when they're about to die because there's no water around. When there's water around, they flourish again. Some of you on your hand wash samples found this huge white colonies, kind of awkwardly shaped. That was bacillus, either Bacillus cereus or Bacillus subtilis. Bacillus is spore forming, so this is Bacillus subtilis, spore forming rod. So you see the green endospores. How did it come to your hands? You probably grabbed on something that had dust on it, dry dust. Okay, some dusty surface. I can guarantee there's going to be a lot of spores on that surface. That make sense? It ended up on your hands. You swapped it, put it on the blade, and a lot of moisture, a lot of nutrients. It started to grow back again. And final stain that I will mention is the flagellar stain. First of all, why cannot we see flagella without staining? Why we can see it under the light microscope? Yeah, it's too small, it's too thin. How can we make it visible? What stain does? So we can see it now. Not just colors, it makes something to flagella, it does something to flagella. If it's too small, what does it do? It makes it bigger, makes it thicker. So molecules of the stain just bind to the flagellum, the protein that comprises flagellum, and just makes it thicker. Now you can see. So flagella is not that horribly thick. It's much thinner. We just see it this way. Why it is important to see flagellum? What is the purpose of flagellum? What is movement? Yes. So if bacteria, compared to microbes, potentially infectious microbes, one that can move and one that is possibly carried by the stream, which one do you think may have more profound impact on the host in terms of disease? Like moving, yeah, because it can swim to higher nutrient concentration, okay? And motile bacteria are usually more pathogenic. So again, we'll look at another virulence factor. Now this table gives you a summary of the stains. I'm not gonna review these stains again. Okay, it's really a summary. I just provide you so you can go. People have different learning styles. Some people want it very, very structured, so this is your very, very structured. What we gonna chat about now is how we grow and identify microbes. So before we even get to that, I want to ask you, why do we want to grow them? Why do we grow microbes? Can't we just take and study them? Why can't we just collect somebody's urine and study microbes in the urine? Why we prefer to grow them, or collect somebody's sputum or swab from the throat and study them. Okay, but why can't we diagnose the disease in the just swab of the throat? Well, you collect my throat swab, you put it on the plate, it's unique, it's mine, it's not yours. I mean, it's still going to be different. Why do we put it on the plate? Huh? Okay, so we can growth differences may be of a diagnostic importance. Huh? Why can't we just do all that stuff with a swab? Huh? Well, you can, they're not going to be bigger when you grow them on the plate, but you're on the right track. You're on the right track. So, not that the cells are small. What is small? Huh? Number of the cells. Number of the cells, right? You collect, you have 100 bacteria. Good luck finding them under the light microscope. You put them on the plate, you have all varieties, and you now can see, oh, I have Staph aureus, I have hemolytic streptococcus. You know, you immediately can start identifying. Does that make sense? It helps us to accumulate large amounts so it's convenient to work with, and it gives us some tools to diagnose them while they grow, even before, you know, we separate colonies. Does that make sense? In culturing microbes, 4 or 5 I's. So first, you have to collect sample and inoculate the medium. Second, you have to incubate your medium in the incubator. Next, you may need to isolate the colonies. Then you inspect whatever you've got, and you identify which microbes you have. So we're going to talk about each step separately, and what do we do during each step. First, inoculation. In order to inoculate something, you have to collect the sample first. What is probably the most important thing that you have to think about when you collect sample? Sterile, yes, sterile collection. When you collect patient sample, swabs that you used for collecting sample from your hands are sterile. Syringe that you used to take patient's blood is sterile. The cup that you collect urine, well, not you, but patient collects urine into, is sterile. Makes sense? So there's no contamination. There are many examples of specimens that you can collect from the patient. Along with the urine and blood and skin samples, what else can you think of? Stool. You can collect stool. Saliva. Say again? You can collect vomitus. Yes, you can collect vomitus. Actually, great source because you don't have to do endoscopy to probe for kind of a rough, but great source to collect something from the stomach. Are there any other liquids? Well, we got blood, we got urine, we got saliva. There you go, CSF. One of the major diagnostic samples for any type of CNS disease, infections of the nervous system. You can collect tissue, so pretty much you can collect anything. Okay, you have to be mindful, about how this specimen reflects what's really going on. Like with vomitus, it's going through a lot of places on its way out, so you have to keep it in mind. With feces, you have to understand that some microbes in the intestine are more likely to leave your body than others. It's not just hypothesis, my hypothesis. It is known that the microbial community that's in your intestine is slightly different from microbial community in feces. So you just have to keep it in mind. You can collect samples from environment. soil, water, poop. Some of the samples in the environment are pretty interesting. There's a lady in, I think she's at Kent State. She studies microbial communities of caves. She goes into cave, collects. What's interesting about it is that, first of all, caves should be fairly inaccessible, right? Because if humans go there back and forth, what you're going to find there? Human microbes, right? Second, you must not contaminate the cave, which means you have to take all your waste with you. And I don't mean like paper wraps of cheeseburger. I mean biological waste. So your poop and urine have to go into the containers and you have to take it out from the cave to avoid contamination. It's pretty interesting. Once you collected the sample, you inoculate medium. We've already done that with a solid medium, with agar plate. You can use liquid medium. You've seen liquid medium on Monday when I inoculated the bench here with Serratia marcensis. It was microbe that grew in the liquid medium. Okay, liquid medium can you make liquid medium at home? What do you think? Any ideas? Huh? How? Think about anything that would support growth of microbes. Water would not, no nutrients. Huh? So you want to, well, yeast are microbes. No, I mean just sterile media. You can get it at home? I get it at home? Yeah. Huh? Already has microbes. Well, food is the medium. It's not necessarily liquid, right? But if you like make a chicken stock, that's your media. Microbes can grow on it. Not all of them, but some of them will. I mean, you buy milk in the grocery store. You buy media with microbes in it already. It is pasteurized, of course, but come on, leave it outside for a couple of days and you will see it has microbes. So media is anything that would support growth of microbes. So liquid. We even use live media, like eggs, okay, or animals. Some microbial organisms must be grown in animals. We have no other meaning to do that, okay. So medium is not only allowing us to grow microbes. In many cases, it helps us to identify microbes. Going back to what you proposed, how it works, how we identify microbes. It can be classified, of course. I always say scientists are people who like to put things in different boxes. I always want to classify things. You can classify it by physical state, by chemical composition, by what it does, okay? And of course, this classification is probably most important because that is relevant to your purposes of the analysis. First, we're going to chat a little about physical state and what are advantages and disadvantages of each type of medium. You already worked with a solid medium, and that's what we're going to do the most, solid medium, the agar plate. This is not a very frequent sample, you see. The agar in the tube doesn't really make much sense. We're going to use the plates, the petri dishes with agar in them. The huge advantage of agar plate is that bacteria, when they grow on agar plate, what do they form? Colonies. They form colonies. So if you have more than one type of bacteria in your inoculum, you can separate these types based on the size, appearance, or color of colonies, right? As we saw, yellow and white different shapes in your hand wash samples. Bless you. Okay? Now this medium is, how do we make it? Take components of the medium, dry components like powders, throw them in the flask, pour some water over it, stick it into the autoclave. Run the autoclave. We have autoclave in that prep room, it's behind that wall. Pretty much a big pressure cooker. Okay? Everything melts because the temperature is high. It's about 250 Fahrenheit in autoclave, and pressure is about 2 atmospheres. When it's done, you take it out, and if you don't have to add anything else to the medium like antibiotics or something, just put your Petri dishes and just pour it in Petri dishes and leave it outside, and it solidifies as it cools down. Okay? If you heat it up, it melts. If you cool it down, it becomes solid. Does that make sense? Such media is called liquefiable. I'm not sure that I can spell it right, so don't count on me. That's probably the spelling of liquefiable media. I mentioned already that you can buy agar at the grocery store. I know that, especially for like science fair in the 4th, 5th grade, some kids use Jello. They buy gelatin, make liquid gelatin, let it cool down. It makes perfect Petri dish actually. The only problem with gelatin is that some bacteria can actually eat gelatin. They can destroy it, use it as a nutritional source. So if you accidentally find that type of bacteria and put it on a Jello plate, Jello will be gone and bacteria will be gone with it because they will die of starvation eventually. Probably looks like fun. Liquid. I'm going to talk about semi-solid last. Liquid medium, on the opposite to solid, is liquid at the, basically room temperature. Anything that does not freeze at the freezing temperature. Okay? Water-based. We don't make medium based on alcohol or acetone because microbes don't really like those chemicals. They die. Okay? And it contains various nutrients. Usually, you can use milk, as I mentioned, it's a liquid medium. You can use chicken broth. There are other nutrients that can be used. Extract of beef, pretty much a powder. Yeast extract, partially digested soy protein, tons of different nutrients. And the advantage of the liquid medium, you can easily add some indicator to it. Indicator that would, for example, change pH. Make sense? So say you have a microbe that digests urea. The product of digestion is ammonium, and when you add ammonia into the medium, which pH ammonia solutions have? Any ideas? Chemists. Is that basic or acidic? Ammonia. Huh? It's basic. Yes, it's basic. So it's ammonia is slightly basic. Okay? So you see how media that was not inoculated becomes basic, and indicator, which is probably phenolphthalein in this case, changes the color to pink. So you can say, okay, my sample had microbes that digest urea. When I say statements like that, good question to ask me or yourself. So what? Where would you find urea in the human body? Huh? Urine. And where is urine? Where does it flow through? Urinary tract, right? Do people get infections of that tract? Oh, sure. And of course, microbes that infect, a lot of them, evolved and acquire the ability to digest urea, which is the good source of nitrogen for them. Does that make sense? There you go. So you can say, okay, those are urease positive microbes. Likely this dude has a urinary tract infection. Okay. Now semi-solid, clot-like. What I mean by clot-like, if you have a tube with semi-solid medium and you tip it over, it's not going to pour out like liquid. But if you hold it down for a long time, it will slowly crawl down. Does that make sense? Like a gel. It's not entirely solid. There's no like solid surface. Why do we use semi-solid medium? Liquid medium. Microbes, so okay, to distinguish between microbes who can and cannot move. Okay, you cannot distinguish them in the liquid medium. They will be just moved around by diffusion of Brown's movement. Okay, in solid medium, it's too solid. Microbes have hard time spreading out. In semi-solid, it's really convenient. So you can see this guy is not inoculated. You have transparent medium, and here you see the trace of the media being inoculated, not by the wire loop, but like a needle. Okay, so needle went in. You see microbe, whatever it is, did not really go anywhere from the site of inoculation. So it's not motile. It cannot move. Here, medium is cloudy all across the board. Every part of the medium has microbial growth, which means this microbe is motile and can move around. Does that make sense? So motile, non-motile. Now what the heck is number 4? It's black. What it is? Some microbes can reduce sulfur. Essentially in this type of the reaction. Say SO sulfate into sulfide. When there is sulfide produced, if media has iron in it, the compound known as iron sulfide is formed. It's black and insoluble. So it precipitates, gives media a black color. You know it's a sulfur reducing bacteria. Again, so what? Presence or absence of enzymes that can reduce sulfur allows you to distinguish between just different species of bacteria. Does that make sense? It helps you to diagnose things. Any questions on the physical state? Not really. They will probably arise. Composition. 2 major types of media. Yes, go ahead. Plate, yes. And it what? Colonies, yes. So is your question about the size of the colony? Why some colonies are bigger than others? Yeah. And why some are so little? Why? Oh, on the plate. Okay. So first question: why we have like 2-3 species? It turns out that Staphylococcus epidermidis and Micrococcus luteus are 2 dominant bacteria on the skin. That's number 1. They just, in such abundance that they, should I say, overwhelm everything else. Okay? They outcompete everything else, even on the plate. Second, not every microbe that exists even on your skin or in your intestines is going to grow on the plate. Competition, maybe nutritional environments, maybe some minute factors that are, say again, for instance, sebum, okay? Sebum is present on your skin, but not on the plate. Maybe those guys require sebum. Does that make sense? That's why few species. Second question, size of the colony. It's probably genetic differences and gene expression differences between those cells that you have collected. Also, the competition, you notice that if you have just few colonies, these colonies are going to be bigger. If there are plenty of them, they're going to be much smaller. If you would compare those white and yellow colonies, remember you had staph and you had micrococcus of that size, and then you have gigantic bacillus. Bacillus is motile. So when it grows, it starts to spread, just crawl on this, almost crawl on the surface of the agar. Does that make sense? You want a whole bunch? Next week we're going to do the ubiquity study. So we'll go and sample anything you want. And I suggest you think about 2-3 things that you may sample. The reason why 2 or 3 things, first of all, we can divide each plate, so you can do 2. Second, if half of the class samples their cell phones, trust me, it's going to be boring. You're going to find all the same stuff on the cell phones. So be creative, okay? Think about something reasonably available. I mean, if somebody wants to do the rectal swab, it's probably not a good idea, okay? I mean, you can do that, but it's going to be a little bit complicated, okay? Or if you want to go like deep in the forest, well, grab the cotton swab today and do it on the weekend, okay, or right before the class, because you just can't spend 15 minutes walking back and forth to swab something in the forest. But yes, if you do that, I would suggest you put the swab back in that paper thing and then put it in the fridge, not in the freezer, in the fridge, and keep it till then. Some of them will die, so it won't be perfect, but a lot of them will remain. That makes sense? So think about what you're gonna sample. And if you want a variety, bottom of your shoe. It's kind of useless because what you see is like 1 layer of bacteria, 2nd layer of bacteria, and then 3 layers of fungi. It grows like crazy. You have so much crap over there. Okay? But it's kind of fun to watch. Okay? So think about it. So that's where you're going to get a variety. Your hands, not so much. Unless just before that you scrape the bottom of your shoe. Then it's going to be a different story. Did I answer your question? Good. Okay. So media composition. Sorry, what did I just do? Complex and defined. The difference is that complex medium, you don't know the exact chemical composition. In the defined or synthetic, you do know what's in the medium. Look at the defined media here. You know exact amounts of amino acids, vitamins, salts, water, okay? You know all of it. So say if I make this medium and then you make this medium, they're going to be identical. All those things are going to be identical. An opposite is complex medium. You throw in something which chemical composition you don't know. Like the partially digested soy protein, which you can find in TSA, in agar that we use for growth. You don't really know chemically what it is. Or you can take, like here, brain-heart infusion. So brain and heart tissue are lyophilized, made into powder. You dissolve it in water. Who knows what's there? Who knows what chemicals are there? Does that make sense? Even different stalks of a brain-heart infusion from the same company can be slightly different. Now, I usually compare complex medium to a buffet. Even if you're fairly picky, you go to a buffet restaurant, you will find something to eat. Make sense? Defined media is party at your friend's house when they serve chips and salsa. I mean, that's it. Chips and salsa. If you don't eat it, that's your problem. Does that make sense? Now, if complex media has, and obviously, to grow something on a synthetic, you need to know what the microbe needs. You have to know the nutritional requirements of a particular microbe. You have to know that it's going to survive and grow on these concentrations. If it's so complicated, know the nutritional requirements, all that stuff. Why do we use synthetic medium? Why don't we just throw a bunch of crap like yeast extract and soy protein and, I don't know, casein, the milk protein, and some sugar, the water, mix it all up, and grow microbes on it? They're going to have plenty of nutrients. Why use defined medium? Good. What do you mean by control? Excellent. Because it's controlled growth environment. Does that make sense? For example, if I want to see if a microbe can grow without amino acid leucine, I can cut out leucine. Where's leucine? I can cut out leucine, right? Easily. Here, good luck taking out any of the amino acids. You can just isolate them out, right? So that's the idea behind synthetic medium. Finally, functions of media. Media can be of a general purpose. It's usually complex. Like TSA, general purpose means that it's okay for everything. I usually say, when jack of all trade, master of none. Okay? It's okay. It works for everything. It's a first choice usually if you want to grow your sample, like sample from patient or sample from the environment. So it has nutrients that would likely support growth of pretty much every microbe. Enriched media supports the growth of so-called fastidious microbes. So what is fastidious? In human language it means picky. Fastidious microbes require some component in the medium. Usually this component is called a growth factor. Picky. Like, well, now picky, in humans, picky is usually defined by allergies, right? I mean, my son has allergies, so I'm not offending people with peanut allergy or gluten intolerance. But, if they serve only bread, and if you have gluten intolerance, you're not going to eat it, okay? For microbes, it's opposite. They have to, the fastidious microbes have to have something in the medium, must have, to grow. And one of the best examples of fastidious microbe is the microbe from the genus Neisseria. Anyone knows what these guys cause in humans? There are two diseases, different diseases. One is here, one is here. Well, they're different species, same genus? Huh? Meningococcal infection, Neisseria meningitidis. Neisseria gonorrhea causes sexually transmitted disease, gonorrhea. They grow on the media that is called chocolate agar. It has no chocolate. It's named for color. It is chocolate because when it is prepared, the cooked blood, like the heated up, blood that heated up with red blood cells being destroyed, is added to the agar. Okay? So cooked blood gives it a chocolate color. Another example of enriched media is the blood agar. Blood agar, unlike chocolate agar, contains fresh blood. It's not human, it's sheep blood. So red blood cells remain intact in that agar. Some microbes can destroy red blood cells. Completely, just destroy them. And you can see this hemolytic microbes here. What happens is around the colony you can see this circle of absolutely clear area. No blood in this area. They completely destroyed the red blood cells. Other microbes will not destroy red blood cells. Which ones do you think are more pathogenic? Hemolytic, that destroy red blood cells, or non-hemolytic, that do not? Yeah, hemolytic, the ones that destroy, obviously, because if they get in the bloodstream, you're screwed. Red blood cells are gone, right? Now I have a question for you. Blood agar and chocolate agar. Both of them have blood as the component. In one case, blood is sort of intact, and another one, it is cooked. What is the precious part of the blood? What is so unique? What is so awesome about the blood? Which component of the blood is so much needed by bacteria? No. You were... narrow it down. Iron. Okay? Iron. Microbes are absolutely love it. They are loving it. Like we are loving McDonald's, they are loving iron. In fact, when we run a fever, what happens to the iron levels in the blood? Anyone knows? It drops. Huh? You're not anemic, but it drops. The reason? It becomes less available for microbes that can potentially go into the blood. Does that make sense? It is a protective mechanism of your body to reduce iron availability when you have infection. It's pretty neat. Now, when you grow microbes on in any type of medium, you may have three different types of cultures. Culture can be pure or axenic, like on this image, the pure culture of E. coli, pure culture of Micrococcus, and pure culture of Serratia. Pure means you have only one species, strictly. Another example of a culture is mixed. You expect more than one species. When you collected samples from your hands, you didn't expect to have only one microbe, right? So that's mixed culture. When you will collect specimens from environment, again, it's going to be more than one. It's all going to be mixed culture. You expect to have more than one. Like in this picture, that's white E. coli's and yellow Micrococcus. Now, contaminated is when you expect to have one, and there is some intruder, like in this image, that you don't expect, which means your culture is not pure anymore. And we want to avoid contamination as much as possible, because it skews the results. see, we're gonna chat about select. You know what? Let's do the review, because I know that it's hard to